So switching gears, almost forget QSAR, and talk about what I think is one of the most exciting development, overarching, almost as exciting as ChatGPT, which I will allude to. And that is a concept that emerged really in the last decade and has taken off enormously rapidly, and that's the concept of biomedical knowledge graphs. Again, the word graph, and how those can be contemplated, conceptualized, built, and used to enable discovery. And we're in the very beginning of the path, so not too many publications. But if you use PubMed and type, you may do it now, biomedical knowledge graphs in the search window, you will see an extremely rapidly growing number of publications on that subject. So I felt that it would be a good time for me to share my excitement about this project, or, if you will, lifestyle. So key points. We're talking about capturing in a mineable format what we could regard as connected knowledge, integrating data, as it says here, from bench to data to knowledge to wisdom, which is the overarching concept of data science in general. Collect data, organize, process, mine, make predictions or inferences, which in my mind equates with the word wisdom. And so we'll talk about knowledge sources, ontology, briefly approaches to how we could mine knowledge graphs and applications, and I will ask you to do an exercise jointly with me, and hopefully you will continue to run those exercises. Because again, I want to share my excitement about doing anything, building and exploring knowledge graphs. I think it's almost unthinkable, at least for me, was to quote a political person, because we're doing science. And amazingly, of all people, Donald Rumsfeld produced a sort of conceptual statement which is extremely applicable to knowledge discovery. And in one of his speeches, he defined 3 major types of knowledge and awareness of this knowledge, which maps onto knowledge graphs exceptionally well, in my opinion. And this is known knowns, known unknowns, and unknown unknowns. And if you think about scientific pursuit and the objective of that pursuit, sometimes we want to validate what we already know. We are in the city of Hershey. We could deduce this by mapping my body onto the map, or all of our bodies onto the map. So we know this. We want to reaffirm. Known unknowns, that essentially means that we know that there are drugs that treat diabetes, but we may not immediately remember what those are. Most interestingly is unknown unknowns, which is when you make unexpected inferences, create de novo hypotheses, or when we deal with problems that have not been defined. Example is COVID-19. We knew about viral diseases, but so more specifically, and kind of start introducing the concepts, factual information can be easily retrieved. Here are a few examples of known information about known drugs and their mechanisms of action. And people working in this field would know this by heart. But look, and this is sort of a hint, how we formalize that knowledge. We have the name of the drug, we have the name of the disease or condition, and we have a link between them, and the link has a name, right? So we have a node, another node, and then an arrow or an edge, labeled edge connecting that node that is verbally described, and the whole triplet has a meaning of connectivity and specific type of connectivity. That's known-known. Known unknowns is you ask a question. You can go into drug bank and find out what drug treats what disease, but you may not know how it's done. This knowledge is in the literature, is in the biochemical pathway books, monographs, textbooks. So you could retrieve that knowledge by asking a different question, right? And then what is known unknown in this case? Known to the world, unknown to you. And that's the example of, can we forecast not only a new disease, which people are trying to do now, but also the place of occurrence and the treatment for that disease. So that's unknown. Several years ago, this concept of knowledge graphs had appeared, but first, before I show you, Google introduced it, why it resonated so easily with a cheminformatician or a mathematician. So I said there is almost no connectivity with QSAR. Not quite. Here is a molecule. And I mentioned in the first lecture that when a cheminformatician sees a drawing like this, and like chemist who looks for places to modify potentially sites that can be destroyed or to which we could add a functional group, we see a graph, and we'll label this graph, and then we compute chemical descriptors. But you could also describe a chemical molecule as a way of capturing knowledge about chemistry, similar to the examples I just introduced when I said drug treats disease. So we have an entity. This entity has a name, so it's a semantic entity. And we have some knowledge associated with this entity from our knowledge of chemistry. And then we have a relation between entities. So a chemical molecule represented as a chemical graph can be used to calculate chemical descriptors from the graphs, graph invariants or various counts associated with labeled graphs. Or you could recognize that it captures the knowledge of chemistry in a knowledge graph relationships where nodes have the name, edges have the name, relationship between nodes is embedded in this kind of information. And if you look at related information about drug-disease relationships, you could represent that knowledge by a small graph or a semantic triplet. And it actually also hints toward making inferences, because if you know that tamoxifen is an antagonist of estrogen receptor and that receptor is involved in the etiology of breast cancer, you could deduce. It could be a knowledge, it could be deduced knowledge. You could deduce by implication that a drug may treat the disease, and that would constitute a potential hypothesis that results from biochemical, medicinal chemistry, and pharmacology studies. So you could connect this knowledge and then make inferences. So that basically, this kind of considerations led to Google at some point recognizing how do we connect knowledge, enable Google searches, which apparently I cannot do here on AT&T Wi-Fi, but other people can. And represent mineable relationships. So when we ask a question, we could go to underlying knowledge graph and mine it to quickly extract connected knowledge in response to a query. And so once we have a graph, we have ways of exploring it, and we have ways of generating connected knowledge, as this example illustrates. And you could recognize that there are various entities that are connected to each other, such as you want to know who painted Mona Lisa and where it was done and who the painter was, et cetera, et cetera. And it's all connected knowledge all around a single node called Mona Lisa. So what is happening? What is this concept of knowledge graph? We typically belong to different disciplines, and within each discipline there are specific concepts. And then each discipline, I just showed you the starting data matrix for QSAR. And that is a knowledge restricted to the area of chemical genomics. We have chemicals, we characterize them, and then we test them, and we store the results in tables, and the tables go into a database such as PubChem or Campbell, and then we mine data from that and build QSAR models. But if you think about what else you want to know, assays are done, and they involve certain biological targets, and these targets are parts of the pathways and live in cells, and you could start asking additional information about the mechanisms and how an assay is related to a disease or toxic event, and that information would typically be stored in another database coming from another layer of knowledge. So if we think about individual data sets and databases that are field specific, but also recognize that there are relationships between entities stored in the databases. CAS is a chemical database, but chemicals have biological activity. If you want to know that, you need to go to Campbell and PubChem. And if you want to know more about the function of the proteins, then you need to go to gene ontology, et cetera. And so the idea is to break out of this data matrix by realizing that objects that are stored within each of these data matrices and within each layer of knowledge, disciplinary knowledge, are interconnected, right? Chemical X is similar to chemical Y, or chemical X can be generated from chemical Y by a particular reaction. That's intra-chemical knowledge. And it interacts with the protein, and protein is stored in different data, or information about the protein is stored in the different database. So you have intra-layer and inter-layer relationships. And if you capture these relationships, you'll create a graph-looking structure. And if you realize that each object in this structure, in each age, have unique annotations, then you have captured the world knowledge in any discipline or across the entire disciplines in the form of organized, structured, annotated, uniquely annotated, mineable knowledge that allows you to answer questions, but also make hypotheses. So knowledge graphs are defined as a way of integrating disparate data that individually follow certain name and nomenclature and rules of the relationships. And the basic unit of, just as atom is the basic unit, when we talk about molecules, the basic unit of a knowledge graph is what's known as semantic triplet. So you realize each node has a unique name, it's an annotation. Each edge has a unique designation or annotation, a label. And in order to form an elementary relationship, you need a semantic triplet, object, subject, predicate. And those concepts come from semantic analysis and from natural language processing, which is behind the concept of knowledge graph. So that's what the elementary unit is. And then you could imagine how you could be building connected semantic triplets based on known or computed relationships between individual objects, and that can grow, and amount to a really structure that together captures this entire world of knowledge. In biomedical research, this concept had emerged as a result of maybe not so rare case of an ingenious funding solution by the NIH and specifically by the National Center for Alternative Translational Medicine that Chris Austin has led until about 2 years ago when he switched from NIH to a private foundation. And he contemplated this whole idea of biomedical data translator, which is a way of building a gigantic knowledge graph integrating various layers from the field of biomedicine, chemistry, biology, anything that relates to the human being. And what motivated this whole development was the realization that we constantly ask questions. Some are simple, some are not. What are the new genes related to Fanconi anemia? That's enrich our knowledge. What are new therapeutic candidates for Fanconi anemia? Now, new therapeutic candidates may imply existing drugs or molecules that can be developed into a drug. So it kind of has a suggestive mode and a hypothesis, and ask for a new hypothesis that could be tested. So you typically would go and try to answer this question by Googling it. And in many cases it may work, but you're going to get multiple answers, and then you need to sift through those. If you realize that the answer is hidden in the world of individual databases, and that world can be regarded as a collection of databases or knowledge sources, and when you map concepts in those knowledge sources and individual objects in those knowledge sources, you may realize that the same object may occur in different databases. Just like in chemical databases, the same molecules could be in PubChem, in Campbell, and in multiple smaller data sets. So if you, in an attempt to answer multiple potential questions, identify all these individual databases and require them to be accessible. Basically, they should be digital, they should be accessible, and one should be able to mine them without having to log in every time. So OpenAPI has to be associated to enable getting into the content of the databases. If this little thing here, which is critically important, I mentioned that the same molecule, I've mentioned in the first lecture that we deal with the world of duplicates. Why? Different people give different names to the same entity, right? So it's the same is true of all databases. If you want to integrate, you have to harmonize, just like we harmonize chemical entities. And so harmonization is called ontology. You specify what concepts you want to use in describing different disparate data, and you map everything onto the same concept. So every single gene has one and only one name, regardless of how individual knowledge sources, creators have called it. So that's what's done in this effort. So you capture data from these databases, and you give each object a unique identifier and unique place in the hierarchy of relationships between objects. Good example that everybody knows about but doesn't necessarily realize of an ontology is mesh ontology behind PubMed. Right? Everything is standardized in PubMed nomenclature. So then you get the knowledge graph, and then you could reason over knowledge graph. You could discover semantic triplets or path through knowledge graph, or make inferences about entities in knowledge graph. And inferences would typically be new edges, postulate of a new edge. I intimated in the beginning how this might be done. So that what creates to the architecture of integrated chemical, biological, clinical, et cetera, objects integrated into a unique single knowledge graph. It could be more than that. Why do we need it, and does it make continuing sense in the age of ChatGPT? It's a good question to ask, right? Because we could ask ChatGPT, and you know that you're going to get an answer. And so as much of excitement it has generated, it has also generated a lot of angst among people interested in facts, right? And this is just to think about this, right? Because when we think about knowledge graphs and sort of the concept of integrated knowledge, it is the same knowledge that is integrated behind the ChatGPT engines, right? It's the world knowledge, and it has amazing capabilities, but not necessarily factual capabilities. And remember, this is a generative model, so it could generate correct answer or incorrect answer, just like we could generate correct or incorrect predictions of chemical bioactivity. So this is an example coming from this publication where ChatGPT I didn't even know this term existed, confabulated a fake research paper. So it referenced something that didn't exist because it generates stuff. And generates means accurate or make up. More examples. So it's important, and I don't think that most people understand the distinction between mining knowledge versus generating knowledge, and danger associated with the generative knowledge. As exciting as it is, it's new. Everything that it generates is new, and it may or may not be based on facts. So I'm not going to talk about, it's a very, very tedious multi-year process that led to the development of modern knowledge graphs. But just mention, you want to identify knowledge sources. You want to agree on, people have different ontologies for different concepts. So you want to agree on ontology. You have a process of building knowledge graph with multiple hidden aspects of this. Then you might explore, generate hypotheses, and that leads to the application. And applications are growing. One of the most exciting potential is drug repurposing, because that's the sort of the most immediate question one may ask. What drug may treat an unknown disease? And I'll show you how this hypothesis may be generated. Data should be suitable, publicly accessible, high quality, actively maintained. Because we constantly need to update. People update individual databases, so we have to immediately update the content of the knowledge graph. Ontology, the heart of knowledge graph. So structures that we've built, knowledge graphs that we have built, use a particular type of ontology. I didn't do anything. Didn't know the word ontology has such a power. Should I think typically you disconnect and reconnect, right? Okay. Okay, yep. Always works. If the car is broken, jump out and get back in, and then it may start again. That's the old. So, okay, so that what needs to be done. So inferences, that goes back to a brilliant bibliometrician, librarian named Don Swanson, who was a professor of information and library science at the University of Chicago, and who really started this whole idea of inductive reasoning as applied to linguistic concepts. In knowledge graphs didn't exist, but biomedicine did. And so he basically created a concept known as Swanson's ABC, a triangle where you have concept A, concept B, concept C. For instance, concept A is drug molecules, concept B is biological processes, and concept C is diseases. Then you could induce that if magnesium modulates vasodilation, which is an underlying cause of migraine, then if that is a new knowledge that magnesium modulates this, you could postulate, which is what he did in his 1990 publication, that magnesium could be used as a treatment for migraine, which few years after was recognized and recommended as one of the treatments. So and that was done through co-occurrence of these terms in the literature. So co-citation of magnesium dilation was interpreted as a functional link. He didn't even do experiments. So this is literature-based discovery at its best. And so you could capture then, if you don't have experimental data, you could capture co-occurrences of terms in the literature and interpret this as a link. And that is done in many knowledge graphs. How it could be contemplated and what led really to our team and several others funded across the country by NCATS to develop this integrated biomedical knowledge was a challenge. And the challenge was explain how imatinib may treat asthma. So cancer drug, lung disease, what do they have in common? Probably nothing. But the challenge that we had to solve in order to qualify to apply for a grant, in order to qualify to write a bigger grant proposal and then get the grant years ago. So it was an uncommon exercise. Was to come up with what they called clinical outcome pathway. Create purely by mining literature, create mechanistic pathway that explains potentially how imatinib may treat asthma. And so you first ask the question about what this mechanism may look like, and the modern drug discovery field and knowledge about mechanisms of drug action tells you that in order to have an effect on the disease, we live in the world of target-specific biology and target-specific drug discovery, so it should have a biological target, and target is part of the pathway. Pathway operates in certain cell type, and or tissue and malfunction of the tissue is recognized as a symptom associated with the disease. So if you mine the world literature and establish all these connections individually, which is exactly what we've done, and say here is a PMID that supports a relationship between imatinib and a particular enzyme. So that really was done manually. And so we answered the question. Turned out we answered the question correctly. So we got an opportunity to develop a protocol that we've called RoboCup that I'm talking to you about today. And you could then realize and recognize this concept of clinical outcome pathway as a way of uncovering mechanisms through reading the literature with the help of mining integrated knowledge in the form of knowledge graphs. And so these are examples of how clinical outcome pathways can be defined as a concept and then discovered by mining knowledge graphs, not doing one by one, but asking a question. Here is a drug, here is a disease. Find a path, high scoring path between a node that corresponds to imatinib and a node that corresponds to asthma, and see if this path is relatively high scoring. So this image comes from a paper, that conceptual paper we published last year about how to define clinical outcome pathways and how to map them onto linked processes within the human or animal body. And so, and as I just mentioned, substance, gene, cell, process, anatomical entity, phenotypic feature, disease. So that could be defined, that could be mined to find common mechanisms between different drugs, for instance, and offer a drug repurposing hypothesis in order to understand, maybe discover a novel target that no one thought about because this target appears in a pathway that connects a drug and disease. So we're again in the beginning, and I'm giving you just some initial sort of concepts and exercises that we have used. Pretty substantial attention to knowledge graphs emerged as a consequence of a single publication, early COVID publication, by a company called Benevolent AI. And they said, we've developed a knowledge graph, and that was sort of about 30% into our own efforts in developing a publicly accessible knowledge graph. They didn't make anything accessible, but they said, we've nominated baricitinib as a treatment for COVID. As you know, that effort has been unstoppable. People have been nominating, pretty much every drug was nominated as treatment for COVID and went into clinical trials, so it could have been one of those funny failed attempts. But that was their justification. They basically said, okay, here's the disease. We know that ACE2 is involved in the entry of the spike protein. So what is ACE2 related to? What are the processes associated with ACE2 from the knowledge graph that they have secretly integrated? And by basically looking at the biochemistry associated and linking ACE2 as the front end human protein interacting with the spike protein of the virus, and real world observation that inflammation is one of the strongest attributes of people suffering from the disease, right? So it's a virus, the human disease. They found pathways associated with it and several kinases involved in processes, including one of them involved in cytokine storm leading to inflammation. And it turned out that baricitinib was associated with JAK1/2, and that was sort of the hypothesis that they've generated. And notably, baricitinib was approved last summer as first as in combination with other drugs, and now as an independent molecule that can be used to treat strong cases of COVID. So that's an example of an impactful discovery that was made by a private company. And of course, we asked the question of, can public effort reproduce this? And so since we use the same primary source, knowledge sources, that the answer was that we could essentially replicate that connection using a public knowledge graph. So once the graph is built, then the question is how do we explore it? And this is an extremely interesting area for a graph theorist and for a graph practitioner, for anyone interested in knowledge mining. And one of the most obvious way of exploring the graphs to create generative knowledge. Generative knowledge in this case is postulating a link that does not exist and has not been captured in the literature, right? And from the mathematical standpoint, association rule mining is one of the most obvious approaches that allow you to find clusters of connected knowledge organized in a particular way, linking nodes of certain type. So the overall approach is there are multiple relationships in knowledge graph. The one that we currently have has over 120 million edges and 30 million nodes, or something like this, and keeps changing because we keep refining it and integrating new knowledge sources. And the knowledge sources change. So it's a gigantic knowledge graph. So you really want to kind of look into these patterns and see if they're logical and consistent. And in the case shown here on the right, from COP, mechanistic understanding of drug action, typically genes or target proteins are involved in the mechanistic explanation of drug disease connectivity. So you know that if you have in a knowledge graph multiple instances of semantic triplets when drug is related to the gene and gene is related to the disease, then it's a type of rule that can be postulated to exist to explain drug treats disease relationship, right? So you're looking for this kind of triplets, and then you could expand the types of rules you might be looking for. So there are two aspects when it comes to graph mining: rule discovery and rule application, rule finding. And I'm using the example of treats because we're very interested in probably the lowest hanging fruit, drug repurposing hypothesis, but you want to really nominate highly confident hypothesis, then follow up with clinical investigations. So rule finding, when you ask what kind of frequently occurring connected subgraphs exist that underlie all cases when drug treats disease, triplet is present. So you're asking for different types of connectivity. And then if you identify those rules, then hypothesis generation means that, so first you mine graphs for subgraphs, and then you take a subgraph and mine the graph to match the components of the subgraph, and then you make a hypothesis that drug may treat the disease. So there is more questions that relate to how you score pathways. I want to go to the exercise, so I'll just kind of mention this in Python as to how we do it with RoboCup, which stands for Reasoning Over Biomedical Objects Linked in Knowledge-Oriented Pathway. This is how we integrate. This is the types of semantic concepts that we recognize: molecular initiating event, key event, adverse to therapeutic outcomes. So this is when you recognize this kind of pathways, then want to search for. These are the types of rules, and they need to identify and score them. There are examples of analyzing the results. So here are examples of rule-based mining where we postulate that, for instance, vitamin, or hypothesize that vitamin A treats common word, and then mine the literature if this H was not captured, because what's captured in the RoboCup are known knowledge, known knowns, right? So known knowns are relationships that come from drug bank. So if the relationship is not in the drug bank, it will not be listed as treats, but the literature may exist that says that this particular relationship has been discussed in the literature with some validation there. So if our rule mining ends in a hypothesis about the treats, then we could look in the literature for facts that validate this hypothesis. There are examples when rule-derived treats hypotheses have experimental confirmation in the literature. Of course, work in progress, refinement of terms, refining of scoring algorithms is important. So there are cases that we found when the relationship has been postulated, but the literature support semantically stated the opposite. That is that it wasn't treated, it was actually increasing the severity of the disease. That is to be expected because we are mining first and foremost the functional connection, and then we still can be inaccurate in the meaning of this connection. So this is just to be remembered as you may potentially start exploring it. I'll skip this in the interest of time, but general question, and I'll share the slides that is addressed here, is how to score. And generally speaking, each connection has a particular level of confidence, and then you could think about ways of summing up sequential confidence for each of the knowledge graph components, or use some other. Interestingly enough, what we currently use is one of the chemical molecular indices that was proposed as a way of scoring chemical molecules as a chemical descriptor. So you'll find this in the slides that I'm going to share. But this basically talks about protocols and quantitative metrics that can score a database. One of the approaches that we use is taken from electrical engineering, where you look for a path with the lowest resistance. And so that's one of the protocols, and resistance of each individual element or edge is estimated from the strength of the support, such as co-occurrence of terms in the literature or direct information about ligand-protein binding, for instance. So Kirchhoff index is one of the indices you could find in cheminformatics literature, and that my colleagues kind of embarked on it independently, and as a cheminformatician, I actually got a strong kick out of how strangely things between fields are connected, even at the level of methodologies. So what I suggest you do is explore a tool that one of my students built, and it's called Exemplar, and the link is on the page, so it's very easy. exemplar.mml.unc.edu. And that's the interface that you're supposed to see. So those of you who have laptops could do it. It's on the web. And what we're going to do, the manual will be in the slides, but we'll try to do this in real time. What we want to do is to establish a relationship between drugs and diseases. This interface basically defines for you the beginning and the end of a path that you want to connect. On the top here is information about how many queries you want to run against Robocop, whether you want to use edge information. So use edge information means that we recognize that edges may have specific annotations, and you may or may not want to use it. You may say, I don't care what the edge is. So if you don't care, then you don't say use edges. Get result metadata will come to this. That's something that allows you to get additional information about the answer. So we're going to reduce initially the path length to 0, and the first node is pre-populated just to show the format in which the input is expected. So the input is expected in the form of commercial drug names. We're going to delete all of this, because the default was to find drugs and mechanisms of action for drugs connected to the Alzheimer's disease, and we're going to remove this as well. So we're making this empty. And we're going to keep the chemical entity empty. The question we're going to ask is, what drugs have been registered to treat a rare disease? So instead of disease of phenotypic feature, we're going to say disease. And in the exercise, we are going after a rare disease called Castleman syndrome, because that happened to be and I'm going to intentionally type Castleman, not Castleman disease, and check for whether or not it exists. And it says it's not found. Try instead Castleman disease. So Right now, we want explicit typing of the disease name, so I'm going to say exactly as it says, disease. Check for terms. Now it's found, and now we're going to ask. And we find 41 unique answers, right? And that is chemical entities that have been mentioned in the literature, if you scroll down, in connection with the Castleman disease. Now we're going to make this more specific, and we're going to say treats. What am I doing wrong?
It's good. It's just chemical drugs or treatments. Just select treat.
I think it's, was it there?
Yeah, treat.
Yes, okay. Okay, no node type. We just have treats, so use edges, treats. We're not selecting any node type. So that's all. So now, if we redo this, no, I think I'm doing something wrong. Let's not use edges, sorry. What am I doing wrong in the exercise? No. I think we want treats here still. Yeah. Okay, let's deal with that. I'm just showing the example. So there are various chemicals that are regarded as treating Castleman disease. Oh, I know. What's that?
So reduce the length by 1.
Yeah. No. Yeah, okay. I think we don't want this. Yeah, sorry. So we want this. So that gives us more manageable number of answers. So now if this only gives you the relationship, right? So it's basically it just says here are the drugs that have been stated in the literature as treatments for the disease. If we want more data, so we click Get Result Metadata and resubmit. Now we get information about the literature associated with each specific drug that we might be interested in that you then could follow. Right now, we don't have a way of clicking on it, so if you want to investigate, then you want to export this observation and then go and mine the literature, primary literature associated with this observation. So that gives us a list of the drug. I think the exercise that we have is figuring out now the next level question. Pick up a drug from this list and see how this drug might work to treat the disease. So we're going to pick lenalidomide and type it under chemical entity here, check for terms. And it says that it's found. And now we want to put here H type. We could put anything. I forgot what we have in the maybe related to, or actually we're not going to use edges. We're going to use phenotypic feature, if I'm not mistaken. Okay. And so, or maybe better use gene. Okay. So the question then when I ask is, if we know that lenalidomide is suggested as a treatment for Castleman disease, what are the genes or targets, potential targets for the action of this drug that are involved and mechanistically involved in the disease treatment? And so that gives us multiple answers, each of which we could individually explore and go into the literature and investigate more specifically whether this particular recommendation or observation from the literature that we found by mining the knowledge graph has strong support, weak support. We could then start going into and investigating individual answers in more detail. So these are just examples of how this could be used. And if I go back to the PowerPoint, there is this ultimate suggestion. What you want to do once you recognize this interface and get familiar with it. But you essentially could start asking any questions out of curiosity or in pursuit of discovery that allow you to mine the world facts captured in databases, integrated in knowledge graph, coming to you in the form of subgraphs that may or may not have meaning. That's the end.